To detect VRE, vancomycin-resistant enterococci, faecium or faecalis, we use the same type of chromogenic agar that works the very same way. It's enzyme specific. And we incubate these, however, for 48 hours. So after 24 hours incubation, if it's negative, it goes back for another 24 hours. So the earliest report you're going to get on a VRE specimen is 48 hours to say it's negative. If the organism grows VRE, it's going to be, again, a mauve or a pinkish color. Very clear, very distinctive, and very specific. But it's specific only for Enterococcus faecium and faecalis. And those two enterococci are the only ones that can be VRE. Because this media is new, the manufacturer recommends that we do further follow-up. So if this comes up in 24 or 48 hours, we need one more day to do susceptibility testing to prove that it is vancomycin resistant and that it is an Enterococcus faecium or faecalis. I think the day is coming when that won't be necessary, but at this point it can take up to 72 hours to get a positive VRE. Once we've isolated the organism, we have to determine which antibiotics can be used to eradicate it. And the most common method used by laboratories is the Kirby-Bauer method of susceptibility testing. To demonstrate the Kirby-Bauer method, let's use a Staph aureus. I take a sterile stick and we make a suspension by sampling 2 or 3 different colonies. And the suspension is made in a tube of sterile saline. And this suspension has to be standardized. And for that we use a colorimeter to make sure that it's a certain specificity or a certain amount of turbidity. In this case it's 0.5, McFarland 0.5. Once that is done, we take a straw and we streak that suspension onto a Mueller-Hinton plate that is used for susceptibility testing. And that is streaked out evenly, and then the antibiotic discs are applied. Which discs we put on depends on the kind of organism we're testing. We have trays of discs for gram-positive organisms, for gram-negatives, and antibiotics that are specific for urinary tract infections. These discs are simply blotting paper that has been impregnated with antibiotic. So for Staph aureus, we'll put erythromycin, we'll put on clindamycin, we'll put on cefoxitin, Septra, and vancomycin. And the antibiotic that is in those discs will seep into the media, and this will be incubated overnight at room temperature. After 24 hours incubation, the plates are taken out of the incubator and we have patterns of susceptibility, something like this. So these zone sizes represent susceptibility, and where you don't see zone sizes, like on these two discs here, cefoxitin and erythromycin, that is showing resistance because the antibiotic is not affecting the organism. It's growing right up to the disc. And you can see here that the organism is inhibited by the antibiotic. And what we do is we will measure zone sizes using calipers to determine whether that zone size is susceptible, intermediate, or resistant for that antibiotic. And we really have no choice. Lab licensing tells us which antibiotics to set up for which organisms, because they're the preferred antibiotics that are to be used. For blood cultures, we use a liquid medium, and this liquid medium has a special coating on the bottom. Bottles such as this, aerobic bottle and an anaerobic bottle. Blood is injected into these bottles, and then they are put into a machine like the Bactec 9420. This machine is an incubator, and it is also a detection mechanism. If I open these doors, you're going to see the panels rocking. You see these panels just come down. When the door is closed, these panels rock back and forth. And these pigeonholes are at body temperature. The bottles, once they're inoculated, are put into the machine like this. The doors are closed, and then they're incubated. If bacteria grow in these bottles, then it produces carbon dioxide. Carbon dioxide affects the bottom coating on this bottle. The machine detects the presence of carbon dioxide, and the machine starts to beep, and the wells start to light up. Something like this. Little lights come up telling us this bottle is positive, this bottle is positive. We take those bottles out, we take some of the blood out, we put it onto blood agar, we do a gram stain, and we proceed with the report. Just a few final comments. I just want to stress that the better specimen you can collect for us, then the better results we can give to you. A simple example would be a urine culture. I showed you agar plates. I showed you the liquid media for blood cultures. Urines are just like that. They're a perfect culture medium. And it's so important for you to get those urines down as quickly as possible, and if you can't, then to refrigerate them until they can come down for an accurate report. And finally, don't be intimidated by the lab. You should know us all on a first name basis. We should know you on a first name basis. No question is too silly or too stupid. We want to help you. You're part of that team. You're the evidence collector and we're the detectives, and we should work together. And I think that's just beneficial for both sides. Thank you for listening.
My name's Helen Shaw, and I am an infection control practitioner at Blue Water Health. I'd like to talk to you briefly about the difference between infections and colonizations. As practitioners, we depend on our microbiology department to provide the identification to us of organisms in the specimens that we send to them. But as physicians and nurses and infection control practitioners, it is up to us to determine whether or not we are dealing with an infection or whether we have colonization from the site where that sample was taken. If we have colonization, we want to ensure that we are not treating with antibiotics. Colonization is more of an organism that is more of a hitchhiker in that location where that sample was taken from. It's not causing an infection. It does not need to be treated. If we have an infection, that would have to be determined by the symptoms that we see in our patient. If it's from a wound, we would be looking for redness, we would be looking for drainage, we would be looking for heat from that area. If those things exist, then we would determine it to be an infection, and we would be treating that site, that infection, with the antibiotics that microbiology would have recommended to us from the testing that they did. If it's in a urine, we're looking for incontinence in our patient. We're looking for back pain and a change in function of our patient. Many of our elderly patients, in particular in our long-term care facilities, will have bacteria in their urine if we culture that. That does not mean that they always have an infection in their urine without those symptoms that I just previously described. Most of our elderly women will have bacteriuria, which is bacteria in the urine, and that will be normal for them. It does not affect how they function. It does not make them ill. However, if the symptoms are there, then we do need to treat with antibiotics, but not every positive culture will be treated with antibiotics. We hope you have enjoyed the snapshot you've had of the internal workings of a microbiology department. They are an essential part of our patient, client, and resident care system. Thank you very much.